Log on, tune in, find out. Another good idea from Cambridge.
Great, thanks. Oops. So this is a problem. We have, we have this thing about two species, a species tree with two species, and think of the species tree as demarked by these fat lines. Then we have these gene genealogies of samples drawn from the two populations. And we're interested in probabilities that the gene genealogies, that they match the species tree. And of course, we would like to extend this for more than two species. But first, we want to get a handle on how these probabilities look like for the very simple case of two populations. And the gene genealogies allow these multiple mergers of lineages. So here we have an example of 4 lineages coalescing at the same time. These probabilities, matching probabilities, have been worked out by James and Noah Rosenberg in the case of the Kingman coalescent. When all populations are Kingman coalescent, meaning that you allow only 2 lineages to coalesce each time, as shown here in the B population. So we want to compute these matching probabilities. So here the gene genealogies do match, meaning that the most recent common ancestors for lineages in both populations arise before lineages from these separate populations coalesce. And here we have an instance of a polyphyly, right, where you have the most recent common ancestor, where you have a merger of lineages from A and B before a most recent common ancestor from either of the population is reached. I have this thing. So let's just look at the rates or at the processes themselves. So the Kingman coalescent, I'm kind of assuming, I don't know how many of you are familiar with that. So that means just that you merge, each pair of lineages merges at rate 1. Then you wait an exponential amount of time with this rate. I choose 2 if you have this many number of lineages that are waiting to coalesce, and then you merge 2 of them. So recently there have been introduced these multiple merger coalescents where you allow more than 2 lineages to coalesce each time. And so this is a case of, as in the Kingman coalescent, you only allow 1 group to merge at any time. There are also simultaneous multiple mergers, but I'm not going to talk about those today. And this is the rate at which any k of the lineages merge. So it's like, the rate is like you flip a coin, and with this rate lambda, which is some finite measure on the 0-1 interval, all that show come up heads, you merge those guys. And a big open question is, for any any natural population, what would the measure lambda be? Here it's just represented in the general form. There are 2 population models. So I'm going to talk about these processes in terms of the lambda coalescence that have been derived from 2 population models. And one is a heavy-tailed population model due to Schweinsberg. And you think of a population as its distinct generations population model. So each individual contributes a potential offspring xi, and those are IID numbers. with these tail probabilities right here. And so this is some parameter alpha, and it will feature in the coalescence rates that come out of this process. And then from these potential offspring, you sample the new population just without resampling, just uniformly across. And this condition on the expected value of the number of the potential offspring guarantees that when n is large enough, you always have enough of potential offspring to be able to maintain a constant population size. And so these are the rates that come out of this. And so this is the number of active lineages that are waiting to coalesce, and you coalesce k of them with this rate, and b is just the beta function, and here is this parameter alpha. So another also very simple model is a modified sort of Mora model where you have just one individual reproducing and contributing offspring to the next generation. And so with this probability here, he just contributes one offspring and the parent always survives, so you have just the usual Mora model. Whereas with this probability, it contributes some fixed fraction psi of the total population. And so what comes out of this is that if gamma is between 0 and 2, you get this some k of the B lineages coalesce with this rate. So how do we compute these probabilities? It's really straightforward. We want to compute them exactly, and the first problem we run into is computing the number of lineages that are that come into the common ancestral population, and so so we need to compute this probability that we have j lineages present in the common ancestral population, given that we sampled I at time 0. In case of the Kingman coalescent, we just derive the spectral expansion of the rate matrix, which is straightforward, and then we can compute the probability this way. We can use the same technique when we have a lambda coalescent, but we have to express the eigenvectors in a recursive form. We can't get a closed form representation of the eigenvectors. Another potential way is just to condition on all the ways of going from I lineages to j lineages. There's only one way in the Kingman coalescent, which only merges 2 at each time, but the number of paths increases exponentially with number of lineages to sampled. So this method is not really practical unless we can somehow identify the most likely path for each lambda coalescent. And here are just some sample calculations. Here we have a sample of size 5, and we compute the most likely path for the beta coalescent with parameter alpha 1.01, 1.2, and 1.5. And if for a large enough sample size, we may be able to identify it as in this way. So when the sample size is large enough, you are likely to have a large initial merger followed by smaller merger until you have just one lineage left. And sort of similar idea for this point mass process. But a problem maybe with this idea is that here you have a really sharp peak. So this path here is by far the most likely path. Whereas here the probability distribution of the path probabilities is very much flatter. So you may have to consider many more paths in the case of the beta coalescence than in the case of the point mass process. So here I'm just representing the probability of this reciprocal monopoly when the most recent common ancestor in both populations is reached before there's coalescence between the populations, and it's just convenient to represent it in this way, and it's just computed recursively. We don't derive any large sample approximations, so we just compute it recursively, and that's straightforward. I'm not going to waste time on this right now. In the same way, it's helpful to represent the non-matching probabilities, the paraphyly probabilities, and the polyphyly probabilities in this way. It helps in the calculations. Let's just get to the results. So this is probability of reciprocal monopoly in case of the beta coalescent, and here as a function of the parameter alpha, when all populations have the beta coalescent and with exactly the same value of alpha, and also against the time of divergence. And we see there's very little effect of alpha on the value of this probability, whereas in the case of the point mass process, there's much larger effect. That's probably due to the I forgot to put the samples. I think that this is for sample size 30 from each population. So very small sample sizes that you can compute this for exactly. Here is just how these probabilities behave as functions of first of sample size. So there is equal sample size from each population, A and B, and here again there is very little effect of the parameter alpha, and this is again the case when all populations have the beta coalescent to the same value of alpha, and also here then the point mass process and with the same value of psi. And the point of this one, so this result and this one is that it's the effect of time scale on the results. So when you may have different coalescent processes going on in different populations, and so you will have to be very careful with how you assign time scale to the time of divergence. Whereas if you have different time scales, you may have the probabilities of reaching the most recent common ancestor in each population being either essentially 1 or essentially 0. And then it will be straightforward to calculate the matching or the non-matching probabilities. And so those are just the conclusions. I didn't show exactly results for the effects of parameters on values of parameters in separate populations, but they vary. The probabilities depend much more on changes in parameters in the derived populations rather than in the ancestral populations. And so again, the conclusion on the time scale, that if you have 3 with different coalescent processes, and assigning time scale to the times of divergence will make a difference. And so I'd like to thank the organizers and the Newton Institute and these grant institutions. So if you have any questions.
Questions? So Bjarki, you have.
I can ask a question about the biological motivation for this. What's the what does looking at a multiple coalescent give you that looking at pairwise coalescence doesn't? So what biological scenarios can you address by incorporating that into the coalescent?
If you have, so these multiple coalescents we think may hold for many marine populations, for example, or a forest trees maybe. They have very different reproduction mechanisms than mammals, for example, so this may be appropriate.
Alexi?
And just following on from that last question, so in the standard coalescent, you've got one parameter you can estimate from data, in the simplest case, the effective population size. In this model, you have 2 parameters, I guess. You've got the effective population size as well as psi or alpha, depending on your lambda model. Is that right?
Yeah. So when you have the point mass process, and you say you scale the population size with some constants, and you're looking at different population sizes in that way. Then the population size scaling constants, they cancel out of the coalescence rates, but they do factor in the beta coalescent case.
So, I mean, has anyone looked at trying to actually estimate the parameters?
Yeah. There are papers out there that estimate both alpha and psi parameters.
Go ahead. So one question. So could you model the situation then where your 2 species A and B potentially had quite different processes? So say one is some spawning marine species and one has a different lifestyle.
Yeah.
All right. Well, thanks very much.